Megasporogenesis. Inside the ovule, we had discussed that diploid tissue is present called as nucellus. So, from the hypodermal cells of the nucellus, a primary archisporial cell will be formed. Primary archisporial cell will be formed from hypodermal cells of the nucellus, which is present inside the ovule. And this primary archisporial cell will divide by mitosis to form primary parietal cells and primary sporogenous cells. Primary archisporal cells are diploid, whereas these cells are also diploid because these cells are formed as a result of mitosis. These primary parietal cells which are there will undergo repeated mitosis and they will add new cells to the nucellus on the peripheral side. So, if you take the diagrammatic representation, this is a nucellus tissue. So, one of the cell become larger, that is a primary archisporial cell. This primary archisporial cell will undergo mitotic division to form into two cells. The upper one towards outside is called as a primary parietal cell, this one, and this one is a primary sporogenous cells. Primary sporogenous cell. This primary parietal cell will undergo repeated division to add cells towards outer side, adding cells to the nucellus, whereas the primary sporogenous cell will now act as a megaspore mother cell. This primary sporogenous cell will be acting as a megaspore mother cell. The megaspore mother cell is diploid in condition, and this megaspore mother cell will undergo meiosis. As it is undergoing meiosis, we consider this as a meiocyte. As we had discussed earlier, that any cell which is undergoing meiosis is called as a meiocyte, and this megaspore mother cell will, when it undergoes meiosis, it will be converted into 4 cells. So, consider here this is a megaspore mother cell. This megaspore mother cell will undergo meiosis to form linear tetrad of megaspores. Linear tetrad means 4 megaspores which are formed will be in a straight line. So, 4 are formed, and as we know that this is a nucellus tissue, and this side is considered to be the micropylar end, whereas this side is considered to be the chelazal end. And here we consider this as a linear tetrad of megaspores. And each megaspore is haploid in condition, and the process of formation of megaspores from megaspore mother cell by meiosis is called as a megasporogenesis. Genesis means formation. That is the genesis or formation of the megaspore. So, we call it as a megasporogenesis. And each and every megaspore is a meiospore, because any cell which is formed as a result of meiosis, we call that as a meiospore cell. Now, out of the 4 megaspore cells, the cells which are present towards the micropylar end, that is these 3 cells will get degenerated, whereas the lowermost cell which is present towards the chelaza, that will remain active and functional. So, that can be represented in the form of diagram where only one cell will become larger, which is present towards chelazal end, and on upper side we can see the 3 small cells like structures which are considered to be the degenerating megaspores. Degenerating megaspores, and the one which is towards the chelazal end is considered to be the functional megaspore. functional megaspore. Now this functional megaspore will undergo mitosis, particularly free nuclear division, to form into a embryo sac or female gametophyte. So let us see how the divisions are taking place in the functional megaspore. Let's start with the functional megaspore. This functional megaspore, which is haploid in its nature, the nucleus of this functional megaspore first will undergo mitosis to form into two nuclei. Out of the two nuclei, one is considered to be the primary chelazal nucleus and other is considered as a primary micropylar nucleus. So out of these two nuclei which have been formed, one will move towards upper pole, one will move towards the lower pole, and here the first mitosis have been taken place. And when they move towards the upper side, it appears to be like this. And now after moving towards the upper side and lower poles, so each of the nucleus will undergo mitosis repeatedly twice. So that first it forms into a four nucleated structure. Two nuclei formed on the upper pole and two nuclei formed on the lower pole. This is by further one more mitosis. One more mitosis means this will undergo one mitosis to form two nuclei. This nucleus will undergo one more mitosis to form into two nuclei. So that here a total of two mitosis will be taking place. Two mitosis are taking place, and further the two nuclei which are present on this side will undergo mitosis to form into four nuclei, and these two nuclei will also undergo mitosis to form into four nuclei. So a total of four mitosis will occur now, resulting into 8 nucleated structure. So out of this 8 nuclei, 4 are present on one pole and 4 nuclei are present on the opposite pole. Now out of the 4 nuclei present on the micropylar end, one will move towards the center, and here also from the chelazal end, one of the nuclei will move towards the center of the embryo sac. Now when one nuclei from upper pole is moving towards the center, only 3 are remained on the upper side and 2 are present in the middle and 3 are remained on the opposite pole. Now one which is moving from upper side, we call it as the upper polar nucleus. This one, and this is coming from lower side, so we call it as a lower polar nucleus. So in total, one embryo sac is having 2 polar nuclei. One is from upper side, one is from the lower side. Now in this, if you observe the nuclear arrangement here, we can see 3 nuclei on the micropylar end, 2 nuclei are in the center and 3 nuclei are present towards the chelazal end. So here this is considered to be the structure which is converted into a embryo sac, where we can see the total of 8 nuclei which are arranged in 3 plus 2 plus 3 pattern. That is 3 on the micropylar end, 2 in the center and 3 nuclei in the chelazal end. So here let's start here. First 1 nuclei, here 2 nuclei, that to be here it is coming to 1 plus 1 arrangement, and here we are getting 2 plus 2, that is 2 here and 2 here, and here we are getting 4 plus 4, 4 on the micropylar and 4 on the chelazal end, and here 3 plus 2 plus 3 pattern. So these are the changes that we are observing in the development of the embryo sac or formation of the embryo sac. Now each of the nucleus will accumulate little amount of cytoplasm and cell wall and converted into a completely developed embryo sac or female gametophyte. So this embryo sac, which contains 8 nucleated structure, This type of structure is first observed in the Polygonum divaricatum plant by Strasburger. So, we call this 8 nucleated structure and 7 celled structure as a Polygonum type of embryo sac. And in this one, the embryo sac is developing from single functional megaspore. That's why we call this type of embryo sac or female gametophyte as a monosporic type. Monosporic type of embryo sac, because the embryo sac is developing from single functional megaspore. So, for the formation of embryo sac, how many number of mitosis are taking place starting with megaspore? 1 mitosis, 2 mitosis and 4 mitosis. So, a total of 7 mitoses are taking place, resulting in the formation of 8 nucleated embryo sac. So, already we had discussed that 1 mitosis will result in formation of 1 spindle apparatus. So, as 7 mitoses are taking place, a total of 7 spindle apparati are formed during the formation of 8 nucleated embryo sacs starting from 1 functional megaspore.